Okay, so we've talked a little bit about this a long time ago when we were talking about evolution, but I want to come back to the question of what forces constrain adaptation. Just to remind you of what these are, because they are going to be coming up over the next few weeks. Remember that one of the things that constrains adaptation is trade-offs. When we were talking about life histories, I mentioned that there's an important trade-off between the number of propagules that an organism can have, the number of offspring that it can have, and the amount that it can invest in each one of those offspring. So for example, you may remember from when we were studying sexual selection that a female kiwi can lay an egg that's about a third of her body weight. If she does this, obviously, she cannot make very many of those eggs. If you want to make many eggs, like the bird that laid all these, you have to make them relatively small, and so you can't invest a lot of resources into each one. Possibly that's going to impact the survivorship of your offspring, but it's an unavoidable trade-off. There are a lot of other trade-offs that are due to things like physics. So for example, the form of a wing that is very good for gliding is not going to be the same form of a wing that is good for rapid maneuvering and flight. There are also trade-offs that are related to cost. Very frequently, when a trait becomes completely useless, it tends to vanish from an organism entirely. And this is because maintaining that trait often has some kind of cost, even a small cost, that will be opposed by natural selection. So for example, when you look at organisms that live in caves, what you often see is the same pattern. They've completely lost their eyes, they have no eye structures whatsoever, and they've lost the pigmentation on their body. And presumably this happens because those actually have a cost, that it is favored by natural selection for organisms to lose those traits. But this has been seen many times in organisms that have adapted to caves. This same pattern. And this is, maybe this is one of those phylogenetically independent observations that suggests that maybe this is an adaptive change in all these different organisms. Another physical constraint, constraint based on just the laws of physics, is one that we've already looked at. When we were talking about the wood rats in Death Valley, we talked about surface area to volume ratios and how they change as an organism gets bigger. And this is a theme that is very, very important in the way that organisms are put together and the way they function. I teach a zoology class when I'm at home at my institution in Southern California, and the running joke in that class is that if you don't know the answer to a question, just write down surface area to volume ratio, because a tremendous number of things about the way that animals are put together does relate to allometry. The way that surface area to volume ratios change as an organism changes in size. Okay, because you get these kind of relationships, you see a tremendous number of adaptations arising to deal with the challenge that you get with either increasing or decreasing size. And so, for example, because a larger organism has less surface area per unit volume, what you tend to see in large organisms are, say, flattened structures, like these structures in fish gills, or folded structures. This is a close-up of the lining of your small intestine. It has these little finger-like projections that help you absorb food. Or in parts of your body like your lungs, there are these highly branched structures. These are like the capillaries that run through your lungs, and that increased surface area helps those capillaries absorb oxygen and let go of carbon dioxide into your environment. When we were talking about the wood rats, I also mentioned that as an organism gets larger, its weight increases faster than its strength. And there are all sorts of things that scale like this. So for example, even the metabolic rate of an organism changes dramatically as its size changes. In particular, the mass-specific metabolic rate changes tremendously. As an organism gets larger, its metabolic rate on a per kilogram basis slows down tremendously. One famous example of why this matters occurred when And I mean, I don't know why people decided to do this. It was the 1960s, I think, and lots of interesting things happened in the 60s. But some researchers decided to see what would happen if you gave a, um, a neurologically effective dose of LSD to an elephant. And so they calculated, okay, a 100-kilogram human being, a physiologically effective dose of LSD is this. An elephant weighs this many more times than a human being, and so they scaled up the dose by that much, and they gave this really tremendously large dose of LSD to the elephant, and of course the elephant basically immediately dropped dead. It was a terrible waste of a perfectly good elephant. And it happened because an elephant's metabolism on a per kilogram basis is much, much slower than humans. And so what would be a normal dose of LSD per kilogram on a human completely overwhelmed the system of the elephant because it metabolized it much more slowly. Never feed LSD to an elephant. It never ends well. One of the concepts that you need to know as we talk about living organisms and how they function is the concept of homeostasis. Homeostasis is basically things staying the same within an organism. Sorry, this slide says animal, but it's true for all organisms, not just animals. It's generally to an organism's advantage if its internal chemical and physical states remain constant. But there are two ways of dealing with the fact that the environment around you changes. One thing you can do is you can conform. Conformation means that you've got an organism that allows its internal state to change as the external state changes. So for example, some organisms, when the temperature goes up outside of them, the temperature goes up inside of them. On the other hand, another strategy is regulation. And this is when the organism maintains an internal state that stays more or less the same, even when the environment outside of them changes. And so, for example, when it gets very cold outside, your body stays at roughly the same temperature inside as it would on a day that was warm and balmy. Okay, so with respect to temperature, human beings are mostly regulators rather than conformers. To illustrate the trade-offs involved with these two strategies, I want to take you to a particular environment called an estuary. An estuary is what's found when you have a freshwater river meeting the ocean. And these are very difficult places to live because when the tide is out, what an organism experiences is basically 100% fresh water. Okay, so there's a very low concentration of ions in that water. But when the tide comes in, salt water will go up the estuary, and the water that an organism experiences will become much saltier. And when you're surrounded by and breathing in water, the change in salinity can have a powerful effect on the salinity inside your body. Lobsters deal with this by just dealing with it. Lobsters that live in estuaries are what are called osmoconformers. The concentration of their body fluids changes as the concentration of fluids outside their body changes. Blue crabs, on the other hand, are what are called osmoregulators. Their osmolarity, the concentration of solutes in their body fluids, remains roughly the same no matter whether they're dealing with fresh water or salt water or something in between. Okay? Now it might seem as though regulation is a superior strategy because it keeps internal systems the same. However, there's some problems. One thing is that regulation isn't free. It costs energy. A blue crab, as the salinity goes down, in order to maintain the same concentration of solutes within its tissues, it has to expend a lot of energy. So as the salinity falls and you've got closer and closer to pure water outside of the crab, it has to use molecular pumps to keep ions inside or push water out, or both. Regulation also requires the existence of stuff like these molecular pumps that can actually do the regulating. Okay, these can be cells, tissues, entire organs devoted to keeping the internal environment the same. If you open up the carapace of a blue crab, you'll find that its gills, which are quite large here, extend over much of the bottom of the organism. The posterior gills down in these areas are where most of the ions are exchanged with the environment. And in the blue crab, these have a tremendous number of transport proteins. Those are what are used to keep ions in or push them out as the salinity of the water changes. And building those proteins is not free. It comes at a considerable metabolic cost. On the other hand, I should say that conformation, the osmoconformer strategy, has real costs as well. Because typically organisms function best when their cells and tissues are at a particular set of values with respect to environmental variables like, say, temperature, salinity, and so forth. In fact, organisms that cannot regulate may be completely excluded from particular environments. For example, echinoderms, sea stars, brittle stars, sea urchins, sea cucumbers, are not found in any freshwater body anywhere in the world, despite the fact that this is a very old group of organisms. The reason is that they actually use the seawater that's outside them as their own circulatory system. They bring seawater in and let it ramify all through their body. And that's how they transport a lot of substances throughout their body. Unfortunately, this means that they're bringing so much of the external environment inside themselves that they basically cannot deal with changes in salinity. If you put a sea star into fresh water, it will die horribly almost immediately because all of the ions inside its body will try to get out and all of the water outside of its body will try to get in. And there's no way that it can deal with that because it doesn't have the capacity to regulate. In order to regulate one's internal state, you need to have a couple of things. First, you need to be able to sense the state of the environment, and this can be the external environment. You can sense what's outside, and your body can begin responding to it kind of proactively, or it can be a sensor that's in the inside of the body, and that it tells you that, hey, the conditions inside are starting to change, we better do something about it. We're going to be looking at temperature for most of the rest of this lecture. So let me talk about some of these, or give you some examples in terms of temperature. So for example, you have heat receptors that are not just in your skin sensing the outside temperature, but they're in your brain sensing the internal temperature. And the temperature of your brain is very important. If your brain gets more than a few degrees above its normal temperature, proteins will start to cook, cells will start to die, and you'll have permanent brain damage. This is why high fevers are so dangerous. In addition, you need to have an integrator. Something needs to be receiving those inputs and making sense of them in some way so that it can send out the right commands. So for example, in humans, the hypothalamus contains the hardware that integrates signals about temperature and sends out the appropriate signals to regulate temperature. And of course, the third thing that you have to have is some kind of effector. For all of these sensors and integrators to be useful, they have to be hooked up to something that can actually change conditions to keep that internal state constant. With human beings, the signals that we get that the temperature is becoming dangerous outside or inside will lead us to sweat, for example. So it'll trigger sweat glands. For evaporative cooling, it might trigger metabolic changes. It might speed up or slow down our metabolism. But most of all, it's going to change our behavior. And that's something we're definitely going to be talking about as we go through this lecture. There's some confusion among students often about what positive feedback and negative feedback mean. It's very tempting to think of positive feedback as something that is good or affirming in some way, and negative feedback is something that is bad or critical. So, for example, if you get negative feedback on a paper, what you really mean is that your professor didn't like it very much. But in the context of homeostasis, and a lot of systems like biological systems, positive and negative feedback have a different meaning from that. Okay, imagine this ball sitting on top of a hill, and we give it a little push to the right. What's going to happen? Well, gravity starts pulling on it, and it keeps moving to the right as a result. Okay, this is called positive feedback because a slight change to the right feeds back positively to keep causing more change to the right. Okay, so what positive feedback is it's feedback that amplifies a change in a system. When you think of the term feedback, you may think of the horrible squawking noise that comes out of a speaker when a microphone is brought too close to it. That's another form of positive feedback. The more sound that comes out of the speaker, the more sound enters the microphone. And that has a positive effect on the amount of sound coming out of the speaker, and that has a positive effect on the amount of sound entering the microphone, and it goes around and around and around, with each round adding to, that is to say, having a positive effect on the signal. Okay, negative feedback is like this. You've got a ball at the bottom of the bowl, and if you push it to the right, gravity kicks in and brings it back where it was. Okay, only negative feedback can maintain homeostasis. Negative feedback counteracts changes in a system and tends to bring that system back within a narrow set of boundaries. And practically every physiological mechanism that we'll be talking about throughout the rest of this class involves in some way negative feedback rather than positive feedback. Positive feedback in biology is very rare because usually it sends things spiraling out of control and it wrecks homeostasis. That is not what we want. So in the context of homeostasis, positive feedback is actually bad.